We'll just wait for a few more attendees to come in, join the webinar, and then we'll get started. While we're waiting, so to keep you updated on our news and the events, make sure you follow us on our social media and subscribe to our YouTube channel so you get a chance to view the past seminars as well. Okay. Seems we get a steady increase still, not reaching to a plateau. So perhaps we start, and while waiting, so let me get started. So welcome everyone to attend today's MBI webinar with Professor Cheryl Grady. Before we start the presentation, I wish to acknowledge the people from the Kulin Nations on whose land Monash University is positioned. I pay my respects to their elders, past and the present. Just a bit of housekeeping. So please ask your questions for speakers via the Q&A button. They will be answered at the end of the speaker's presentation. The chat section has been disabled, so questions must be sent through the Q&A. So now it is my great pleasure to introduce our speaker today, Professor Cheryl Grady. Cheryl is senior scientist at Rodman Research Institute and Baycrest, and a professor in the Department of Psychiatry and Psychology at the University of Toronto. She held the Canada Research Chair in Neurocognitive Aging from 2005 to 2018, and she was an elected fellow from the Royal Society of Canada in 2019. So Professor Grady's research really looking into MRI to determine the role of brain network connectivity in cognitive aging. So without further ado, welcome Professor Grady. And it is our great pleasure to have you today to talk about your research. And that hand over to you. So I will stop sharing my screen and you can take over.
Thank you very much. Okay, hopefully everyone can see my screen. Please let me know if you can't or if there's a problem. Thank you for inviting me today to come give this talk. I'm going to be talking about the neural correlates of cognitive control across the adult lifespan. And before I start, I want to acknowledge the three people whose names are there at the bottom of the slide, particularly Jenny Reek, without whom this project would not have happened because she did a lot of the heavy lifting, and also Brennan D'Souza and Julia Barachini, who both acted as research assistants on this project. So if you like what you see, it's because of them. If you don't like what you see, you can blame me. Okay. Having some difficulty advancing my slides here. Okay, that works apparently. Okay, so first of all, I guess I need to define what I mean by cognitive control. And a good example is from cooking a meal. So cognitive control is generally thought to be the process by which we organize and guide our behavior to achieve some goal, such as cooking a meal. So for example, you might need to remember which ingredients that you have put in and which ingredients need to go into whatever it is you're cooking. So that would involve something like working memory or short-term memory to remember all of the pieces that are involved. If you're doing more than one thing, so you've got multiple pots on the stove, you might need to go back and forth and switch from one pot to the other, which is a type of task switching that's also involved in cognitive control. And finally, you might need to inhibit some distractions that happen while you're trying to cook your meal. So all of these different components make up what we call cognitive control. And in the lab, there are a number of different tasks that we can use to to study these components of cognitive control. In terms of inhibition, which might involve not paying attention to distractors or inhibiting a motor response. Excuse me momentarily, I'm just going to move the people I see here so I can actually see my slides a little bit better. No, it doesn't seem to want to move now. So for inhibition tasks like a flanker task or a go, no go are used in the lab to look at inhibition. For task shifting, which is essentially measuring flexibility, we have things like dual tasks or switching between one task and another. Finally, for working memory, it involves updating information and clearing or storing things in various buffers. For things like this, we use list sorting or in-back tasks. So what I'm going to talk about today is some experiments that we've run looking at these different components of cognitive control and a sample of 147 healthy adults between the ages of 20 to 86. And before I move on, I should say something I forgot to say before, which is that these three components are thought to be somewhat separate in terms of cognitive control components, but they're also correlated with each other. So they have unique and shared variants, at least in terms of behavior. And one of the things we're going to look at today is whether this is also true in terms of the neural correlates of cognitive control. So three questions that we're going to address today are as follows. So we know that there are age differences in cognitive control, performance, and brain structure. And so we're going to be looking at how age, brain structure, and performance are related to activation during these cognitive control tasks, and whether there are unique patterns across the three components that would support the separation of these three components in terms of brain activity, and how they're related to these other things. We're also looking at the components of cognitive control. We know they become less distinct behaviorally with aging, and so we want to know whether the brain networks and their functional connectivity also show this type of less distinct patterns, or something which is also known as dedifferentiation, or activity patterns being less differentiated across the three tasks with age. And finally, we've been interested in looking at variability of the Fmri, or the bold signal. We know that decreases with age and is also related to cognitive performance. And so we'll be looking at whether or not bold variability changes with cognitive load and age during our working memory task, which is the one task we have where we have both changes in load and can look at age as well, and whether or not this influences performance on working memory tasks. So to start off, just some general info on the experimental design. We had two sessions. In the first session, the participants came in and did some neuropsychological testing using an instrument called the NIH Toolbox, which assesses cognition over a variety of different domains, such as emotion, motor function, executive function, or cognitive control, etc. And down here, you can see that our sample shows pretty much no change with age in crystallized intelligence, but a reduction with age in something which we call fluid cognition. And this is something that's been shown many, many times before in the literature. So this just shows that our sample is quite standard in terms of the kinds of behavioral changes that you see with age. In terms of the task components, or the Fmri components, this is an example of the inhibition task, or the go, no go task. When there were participants saw many X's and they had to press a button every time they saw the X. They saw any other letter which was infrequent, they were supposed to withhold that response. So that's inhibiting a prepotent response. We also had what we called an initiation condition, in which there were more no-goes than goes, and so the prepotent response was not to respond. Over here you can see the performance. This is a measure which combines accuracy and reaction time, and you can see that there is a decrease with age, as you would expect on this inhibitory task. The second task we looked at was a task switching design in which participants carried out 2 tasks on letters, either determine whether it was a lower uppercase or a vowel or consonant, and sometimes they would have a trial that repeats, so the task didn't change, and other times there was a switch. Whoops, sticky fingers. There was a switch in what the task was, and generally when you have a switch like that, people slow down, so there's a switch cost. You can see that over here, people did have a switch cost, but interestingly, there was no age difference in this sample in terms of the switch cost. And then finally, the working memory task was an in-back task, again, using letters. And we have the sort of the typical 0, 1 back, and 2 back conditions. And you can see over here that the accuracy declines as the task gets harder, as you would expect. And that decline is even larger in the older adults than it is in younger adults. So to look at the brain networks, and what I'm going to show you today, we used a particular parcellation, which is from a 17 network parcellation published by Thomas Yeo a number of years ago, and then updated by Schaefer et al. In 2018. You can see that with the different color codes there, the number of ROIs, there were 100 nodes altogether. The different networks that we looked at, you see over here on the right. So it includes things like the dorsal attention network, the ventral attention network, the frontal parietal control network, which some people think is the main network for underlying cognitive control, the default mode network, and a limbic network. I will just point out that there were also some regions which will be in future graphs labeled as unknown, which are not covered by this parcellation. These include some subcortical regions in the hippocampus. So when you see something unknown, it's basically those subcortical regions and including the cerebellum. And so today we're going to be talking about a variety of imaging measures that we obtained from our data set, including task-related activity and functional connectivity, task-related bowl variability measures, structural measures primarily in terms of cortical thickness, and white matter measures from DTI, primarily functional anisotropy, of various white matter tracks from tractography. So, question 1. How are age, structure, and performance related to activation during cognitive control tasks, and does this relation differ across the 3 components? So just as a bit of background, we know there are age differences behaviorally in these 3 cognitive control components. So in young adults, if you look at a set of tasks measuring these 3 components and do something like a PCA analysis, you'll find that the 3 are generally fairly distinct in terms of their loadings on these components. Whereas in older adults, they tend to be a bit more overlap, so they're not as distinct in terms of their variance components as you would see in a young adult. We also know that there's brain loss and aging, both in gray matter, which you see here on the left, and white matter on the right. This is data from a longitudinal study where people were assessed up to 5 years. And you can see this is a volume measure including both gray and white matter in these various lobes. You can see that there is a decline with time in older adults, and both gray and white matter, and it's fairly widespread in terms of both. So knowing that there are these changes in behavior and brain structure, what we wanted to do was to try and model brain activity during these cognitive control tasks and see how it relates to these different factors that I've just mentioned. So essentially what we did, we used something called partial least squares, which is a multivariate analysis. And we modeled task-related brain activity across the 3 tasks in the same analysis, so we could look at similarities and differences. And we modeled it as a function of age, FA, and thickness in frontal parietal regions, thinking that these would be maybe the ones that are primarily involved in our cognitive control tasks, and task performance. So these 4 variables were used to model task-related activity. And with PLS, like other decomposition techniques, you get latent variables or components. And in this case, they consist of 2 parts. One is some areas of the brain that show an effect, and then the effect itself. So in this case, it's going to be correlations between one of these 4 variables and scores which represent activity in these various areas above threshold that you will see. So the first latent variable or component from the analysis showed correlations with factors from both the inhibition task and the set shifting task, but not the working memory task. So in this case, performance and FA were correlated on the inhibitory task, and age and brain matter or thickness, cortical thickness, were correlated on the task switching task. So this generally is a component which shows differences in that its activity in these yellow and red areas is related to performance on the inhibition task, but not on the task switching task and not on working memory either. Whereas one commonality is that there are negative correlations between activity in these areas and both gray matter measures and white matter measures in these 2 tasks, such that greater thickness or FA is associated with less activity. And this type of relation has been interpreted by others and by ourselves as indicating that the better the white matter and gray matter structure is, the less you have to activate these regions. So it's sort of an efficiency. It serves as a scaffold for more effective or more efficient brain activation. And the regions that, or the networks that mainly are showing this effect are in the default mode network, which you see here, the frontal parietal control network, a little bit of some of the other networks, and then a lot of the so-called unknown regions, sort of subcortical regions, which medial temporal areas, which you can see here in subcortical regions also are showing this effect. In the second latent variable, we again see some differences and some commonalities. So this particular pattern of effects is correlated with age in all 3 tasks, but in the inhibitory task, older adults have less activity in these red areas that you see here, whereas during working memory and this task switching task, they have more activity in these areas. Performance is related to activity in these areas. In inhibition and working memory, again, more activity in these red areas is associated with better performance. And only in set shifting is activity in these regions related to cortical thickness. So again, we have some similarities, but some differences across the 3 tasks. And the regions that you see here in red are mainly from the default mode network. So the fact that this is related to performance may actually suggest that during the inhibitory and the working memory tasks, if you essentially have more activity in this default mode network region, which is supposed to have reduced activity during the task, then maybe you're finding the task easier and you have more bandwidth, so to speak, left over to activate your default mode network. At least that's one interpretation. So to summarize, we found both common influences of age, structure, and performance on brain activation during cognitive control tasks, but also differences. In terms of age, we found that task-related activation increases with age for shifting and working memory, but decreases for inhibition. And task shifting showed the most widespread correlations between age and brain activity, showing such correlations on both of the latent variables. In terms of brain structure, for shifting and inhibition, increased thickness and FA and white matter were associated with less activation, consistent with the idea that structure may provide a scaffold for efficient activation in various brain areas. Interestingly, structural measures were not related at all to working memory activity after accounting for age and performance, which was somewhat surprising to us. In terms of task performance, increased activity is related to better performance for inhibition and working memory, but this relation is particularly widespread in the cortex for inhibition. And shifting performance was not related to activity in any of these identified areas after accounting for age and structure. So essentially we find that these results extend the three-factor model of cognitive control to the aging brain by showing that the influence of age, brain structure, and behavior on brain activity does depend, at least in part, on the specific component of cognitive control. So there's both shared variance as well as unique variance. Now moving on to question 2, we're going to be looking at whether brain networks show age-related dedifferentiation across cognitive control tasks in terms of their functional connectivity. So we know that brain activity can be dedifferentiated or less distinct in older adults across a number of levels. This example is on a processing level. So this was some data we collected during encoding tasks, encoding for specific items versus encoding for associations made between those items. And in this particular set of regions that you see here in red and yellow, younger adults showed more activity in these brain regions during associative encoding than during item encoding. Older adults show no difference at all, consistent with the idea of less distinct or less differentiated patterns during encoding. But this kind of pattern has also been seen at a more basic level of stimulus representation. So this is a recent study published in Journal of Neuroscience in which younger and older adults viewed pictures of faces in buildings. And in these occipital areas that you see highlighted here, both groups showed greater similarity within items. That is, faces were more similar. Brain activity during faces is more similar to other faces than it is when you compare faces versus buildings, for example. So within items, similarity is much higher than between items similarity. And you see the same thing in older adults, but the distinction is, again, much less in older adults, suggesting that even at more basic representational levels, older adults have dedifferentiated patterns of brain activity. So what we're going to be looking at here, again in these networks, just to remind you of what they are, is patterns of functional connectivity across our adult lifespan sample to see if the patterns of functional connectivity also are more dedifferentiated. So this is a rather complicated slide, but it summarizes the method that we used here, something called Cove status. And essentially we took data from all of these 100 nodes in each of our participants and created functional connectivity matrices like you see here. And the first thing we did was to look to see whether the functional connectivity similarity across all the networks and across all the tasks was how similar those measures were for 4 age groups. And I'll show you what the age groups are in a minute. We initially divided them up into 4 age groups. So how similar is the connectivity across the working memory, the set shifting, and the inhibitory tasks by age group? So does the functional connectivity become less distinct across the age groups? Then by creating a consensus map across all of the individual participant maps, we then decompose that to determine the multivariate connectivity space of all the various networks in the 6 tasks conditions that we had. And then from that, we can calculate what we call task differentiation scores for each network and each participant. So if this is the mean value for this participant and the connectivity values for all the different tasks are quite dissimilar or widespread in this convex space, then this person is, you can say this person is differentiated. Whereas someone that looks like this, if all the tasks are clustered around the mean and very similar to each other, then that person's functional connectivity is not very differentiated. It could be dedifferentiated. And then finally, for each network, we calculated a segregation score to look at how segregated the particular connectivity within each network might be relative to between networks. So if a network within its nodes has a lot of connectivity within the network, but there's not much connectivity of those nodes with other networks, then we say that that network is quite segregated. Otherwise, it might be more integrated in the sense that it has more connectivity with nodes outside of its core network. And we're also looking at the effects of age and per task on these segregation scores as well. So first, just to give you an idea of what the space looks like, here are the various networks in different colors these diamond-shaped things are the means across the various tasks. This axis is the first component, this is the second component. And you can see that on the first component, there's a lot of separation, for example, between this dorsal attention network and limbic networks over here, whereas on the second component, there's a lot of distinction between the frontal parietal control and the default mode network with both of the visual networks. So it just gives you some idea of what the network connectivity looks like across all the participants in a multivariate space. So this is this functional connectivity similarity across the different tasks. So we have the initiation and the inhibition task, or measures that come from the go, no go, the task switching, and then the 3 loads of the working memory task. And you can see in younger adults, the working memory and inhibition look rather similar, but they're quite distinct from the inhibition and the set shifting. And then when we get even to the next age group, this is just the 32 to 48, we can see that the inhibition and shifting have started to merge together and they're no longer distinct. This is also the case in the somewhat older middle-aged. And by the time we get to the older age groups, the initiation task is now no longer distinct from inhibition and starting to look like it's going to merge with 0 back. So the dedifferentiation, if you will, begins even as early as the 30s and 40s and is even more pronounced in the older age groups. So now we're going to look at the task differentiation scores per task. Again, this is what a highly differentiated individual might look like, and this is what a low differentiation or dedifferentiated individual might look like. And what we found was that there are some networks which do show a decrease with age in this task differentiation score. So for example, the frontal parietal control network, the default mode network, salience, ventral attention network, et cetera, and even a somatomotor network, although this one was more nonlinear. But these networks all showed a reduction in how differentiated the various tasks were with age. So the older adults tend to look more like this, whereas younger adults tend to look more like this. In terms of segregation, we also found age reductions in some of the networks in segregation, again in the frontal parietal control network, default mode network, and some of the other networks. Interestingly, we found one of the somatomotor networks actually had an increase in segregation with age. Decreases have been reported in other groups, but I don't think anyone so far has found an increase in any of the networks. So this was somewhat unique perhaps to our sample, but generally in line with the task differentiation scores, the network segregation also decreases, suggesting the networks are starting to have more between network connectivity as opposed to within network connectivity. And finally, we looked at the relation between network segregation and behavior. And we found that regardless of age, so we regressed out age from these correlations, there were a couple of tasks that were correlated with segregation. So this working memory task was correlated with more segregation of the control network. So as the frontal parietal control network became more segregated, that was associated with better working memory performance. In contrast, as the dorsal attention network became less segregated, so it becomes more integrated, and that was correlated with flanker task performance that you see here. And there was one that showed an interaction with age such that only in older adults there was a correlation between salience and ventral attention network segregation and switch cost. So only in those older adults, if this network was more segregated, then the switch cost was lower, which means performance was better. So in general, there was a correlation with segregation in some of these networks and performance, but it wasn't always in the same direction. So in some cases, segregation was better for performance, and in some cases, integration was better for performance, regardless of age. So to summarize this section, we found that network functional connectivity patterns during inhibition and shifting were more similar beginning even in the 30s. with additional task overlap in the oldest age group. The task differentiation scores were negatively correlated with age, particularly for the frontal parietal control, the ventral attention, and the default mode networks, providing evidence of dedifferentiation of functional connectivity patterns underlying cognitive control with aging. Networks exhibited age-related reconfiguration such that a number of networks, again, including the frontal parietal control and the default mode network, were more integrated, whereas the sensory motor network, or one part of the sensory motor network, was more segregated in older age. But generally, it's a pattern of less segregation with older age. So essentially, we found developmental differences in how cognitive control is supported by functional connectivity and brain networks across the adult lifespan, predominantly in those networks that are thought to underlie flexible engagement or disengagement of network activity as a function of task demands, that is the frontal parietal control network, or goal-directed attention as the dorsal attention network, or representation of task-related schemas, which are thought to be represented by the default mode network. So finally, question 3, we'll be looking at whether Fmri or bold variability changes with cognitive load and age during working memory, and whether this influences performance. So we've been interested in bold variability in the last few years. And the idea here is that we know that there's a lot of variability in the nervous system, both at all levels, both at synaptic levels, interactions among neurons, larger systems, how the neurons influence muscle, for example. And all of these types of variability generally have been thought to be nuisance variables that one should remove, but it turns out that there may actually be very interesting signal inside some of these variability measures. So in terms of brain signal variability, like from the EEG or the bold signal from Fmri, recent work has been looking at neural variability and how this changes with various things like task demands or neuromodulation from various neurotransmitters, and also how it relates to behavior. So what we're going to be looking at here is, first of all, task demands. So how the cognitive load changes during our working memory task and how that influences neural variability as measured from the bold signal. And also we're interested in interperson differences. In this case, it will be age. So how does cognitive load and age influence bold variability, and does bold variability relate to behavior at all during these cognitive control tests? So we know from our previous work that brain signal variability is sensitive to both cognitive load and age. So in one of our early studies, we used a face perception task in which the faces were degraded by some amount from not very much to quite a bit. And what we found was that these areas that you see here in yellow and orange showed a decrease in variability from a condition where there wasn't much to the condition where there was the most degradation of the faces, suggesting perhaps that as the task gets harder, some brain areas need to focus their activity more and become less variable to meet the task demands. We also know that it's related to age. And what we found in this study was that these areas which you see here in yellow, there are a few of them. There's one in the cerebellum, maybe in medial temporal areas. Those areas showed an increase in variability, bold variability, with age. There was also an increase in reaction time and an increase in intra-individual reaction time, or within-person reaction time variability, with increased variability in those regions. However, most of the areas that we found, these blue areas, which are much more extensive, showed the opposite relationship. So in that case, there was more variability with younger age, and with more variability, there was faster reaction times and less variable reaction times, suggesting that while there may be some areas where increased variability could be found in older adults, it may not be associated with better performance at all in those older adults. So what we're looking at here then is how all this relates to our working memory tasks. And we use two different working memory measures in the analysis that I'm going to show you. One is from the NIH toolbox. So this is a list sorting task, working memory task. You can see that there is a reduction with age in performance on this task, carried out outside the scanner. And our in-scanner task is the in-back task. This you've already seen. This is the accuracy, which shows that that declines as the task gets harder, and it declines even more in the older adults than in younger adults. Over here, we see the within-person variability measure of RT, which also is influenced by load. So variability in performance increases with load across all four age groups here, but the increase is actually greater in the older adults than it is in the younger adults. So there's an interaction with load and age in terms of reaction time variability, and also accuracy in that the differences across load get larger with age. So now we're going to look to see what happens to bold variability. To access bold variability, we do a bit more in terms of pre-processing of the Fmri images that we normally would do, because we want to get rid of as many sources, unwanted sources of noise and variability as we can. So in addition to our standard pre-processing steps, we also do some denoising. In this case, we used the fix module in FSL. We also regress out motion parameters and signal from white matter, CSF, and major vessels in order to, like I said, try and get rid of as much unwanted noise as possible. Then we computed the standard deviation of the bold signal, which is our measure of variability, using the variability toolbox in SPM. We did two different kinds of analysis. One was a univariate analysis to look for effects of load, age, and load by age interaction. And then we also used the multivariate analysis, PLS, to look for relations between SD bold and performance on both the in-scanner task, which was the in-back task, and the out-of-scanner task, which was the list sort task. So first of all, in terms of load effects, we saw load effects on bold variability in rather widespread areas of the brain, which you see here in red and yellow. And like we saw with our degraded faces task, there was a decrease in variability as the task got harder. Again, suggesting more focused activity patterns are necessary as tasks get harder, hence less variable. In terms of our networks, these regions were mostly in the default mode network, although we also saw quite a bit of overlap with the control network and then a little bit of overlap with most of the other networks. But again, it's mostly the control, frontal parietal control, and the default mode network are showing this pattern of decreased variability with load. We also saw a main effect of age in a subset of these areas that we'd seen over there. Again, mostly default mode with a little bit of control network. Here you see the decrease with age. So variability in all of these regions decreases from, excuse me, the younger adults to the older adults. However, qualifying both of these main effects was an interaction between age and load in these areas that you see here in red. And what we found here was that bold variability was modulated by task load. You can see that here, but the modulation decreased with age. So the younger adults had a much bigger modulation of bold variability than the older adults, who had very little. Again, this was mostly in the frontal parietal control network and the default mode network. And just to remind you of what the reaction time variability looked like in terms of behavior, the modulation of modulation of reaction time is larger in the older adults, but it's smaller in terms of bold variability. So less modulation in the brain is associated with a bigger modulation of behavior. And to look at the relation between bold variability and the behavioral measures, we looked at the in-scanner measures were RT variability in our working memory task, in the in-back task, as well as accuracy. And then we also included the out-of-scanner measure from the list sort test to look at whether or not bold variability is related to working memory more in general, since this was collected outside the scanner. And we looked at the relation between these behavioral measures and variability in our three 0 back, 1 back, and 2 back working memory conditions. I would note that before doing this analysis, for the behavioral measures, we regressed out the effect of age, and from variability, we removed the effect both of age and mean frame displacement or motion effects to look at the relations between variability and performance across the lifespan without the effects of age or motion. So the first latent variable that we see here is significant correlations between accuracy on the n-back tasks, seen here in cyan, significant relations between bold variability and reaction time variability, but no significant relation between this pattern of variability and the list sort task. So this was something that was only seen during the in-scanner working memory task. And essentially what this shows is that more variability in these yellow areas and less variability in these blue areas is associated with less variability with in-person variability and greater accuracy on the task. So increased variability in some areas, decreased variability in other areas is associated with better performance on the in-back task and the scanner. And these areas where an increase in variability is associated with better performance was seen primarily in some limbic network areas and default mode areas, whereas decreased variability being related to better performance was seen primarily on the somatomotor task, but somatomotor network, but also in the visual network and some of the subcortical regions. The second pattern showed something similar, except now the correlations are with the out-of-scanner task, the list sort task, and with the in-person variability. And now we see a wider set of areas where increased variability, this orange areas, increased variability in these areas and decreased variability primarily in this blue area here, again, are associated with better performance. So as bold variability in those orange areas increases, within person reaction time variability decreases and accuracy on the task increases. So again, it's accuracy on the list task, list sort, out of scanner task, but also, again, this pattern is also related to reaction time variability on the in-scanner working memory task. Those regions where increased variability is associated with better performance included some somatomotor regions, ventral attention, limbic, and a little bit in the default mode, whereas those regions, primarily this area here, which showed correlations between reduced variability and better performance, was in the visual network, seen here in blue. So to summarize this, we found that aging was associated with increased load-related modulation of RT variability, but decreased load-related modulation of brain signal variability or bold variability, particularly in regions of the frontal parietal control network and the default mode network. So again, increased modulation of behavior, but decreased modulation of brain signal. Decreased with age. Decreased variability in the frontal parietal control and default mode networks with increasing task load suggests a focusing of network activity to meet cognitive demand, and again, a modulation that is reduced in older adults. Beyond aging, so in the sample as a whole, greater bold variability in limbic and default networks and decreased bold variability in visual cortex supported both performance on the in-back task and the list sort task, both the in-scanner and the out-of-scanner task. So supported working memory more generally. So poorer working memory performance in older age may be due in part to a failure of brain regions to modulate their variability in the face of increased cognitive demands. So to conclude all of this, we found that brain activity during the three components of cognitive control showed both common and unique influences of age, brain structure, and performance consistent with the idea that these components have shared as well as unique variants. Network functional connectivity during cognitive control tasks is less distinct and less segregated in older adults, in line with the idea of age-related dedifferentiation of brain activity and functional connectivity. Bold variability declines with increasing cognitive load, but this modulation is reduced in older adults. A smaller load-related modulation of brain variability is associated with a larger decline in accuracy and increased RT variability with age. So a brain that has less variability or less modulation of variability is going to be associated with more variability of behavior. Age-related brain network differences are most prominent in the frontal parietal control network and the default mode network, indicating a key role for these two regions in aging. This, I think, is an interesting point, which maybe we could discuss in the question and answer period. It's not entirely clear why these two networks seem to be showing up in so many different studies, not just in our work, but in other studies as well, as being very important for cognitive aging in general. And finally, these results provide additional evidence that the dynamic range of brain activity and functional connectivity is lower in older adults, which may be particularly important for the ability to use cognitive control for the flexible adjustments that are necessary when trying to carry out goal-directed behaviors. So that's all I have. So I guess now we go to questions, and maybe I need to stop screen sharing.
Great, thanks a lot, Cheryl. It's a very insightful presentation. So now we open for questions. Are there any questions from the audience? If you have any questions, please tap into Q&A button. I guess while we're waiting for people to type in questions, so my background is engineering and physics related to MRI. So when you talk about bold variability, have you considered instrumentation variability, noise from scan to scan, those type of factors? Just wondering whether that play a role or not.
Not directly, although I guess I would hope that the kinds of pre-processing and denoising procedures that we do would account for some of that. I mean, one question that you might have is whether or not variability is related to things like vascular changes with age. And other people's work has shown that if you account for some things like vascular changes with age, you still get age differences in both variability. So I don't think it's just due to things like instrumentation or vascular changes with age, certainly during tasks. Now, maybe during resting state, that's a slightly different story, but I think during tasks, it's influenced by these things, but the age differences are not, strictly speaking, just accountable for by things like vascular and hopefully not instrumentation differences either.
Sure, great, thanks.
Hi, Cheryl.
Hi, Gary.
Thank you for a terrific talk. Wow, that's such a comprehensive program of research you've done. Really terrific and amazing to see these outcomes, looking at the effects of variability on different involvement and of networks with aging. But my question, Cheryl, is really around what is the underlying mechanism, do you think, of variability in the brain, which then gives rise to either improved performance in younger people, but then not necessarily in older people. How do you think it's really working? Do you think that, like, the sort of neural firing rate, if there's more variability, there's a sort of an earlier leading edge to that, and then that could lead through to people having, like, certainly faster response times, but not necessarily more accurate response times? How do you see the underlying neural activity relating to these, the variability of that underlying activity relating to these responses?
Yeah, that's really obviously a difficult question to answer with fMRI. I think, well, certainly the way I tend to think about it, and maybe the way other people tend to think about it as well, is not so much in terms of neurons themselves, but just the ability to switch flexibly between networks. So if you if your network connectivity and activity is, if you don't have a very large dynamic range, then the idea is that you probably aren't able to switch between networks depending on what your cognitive demand is. So, you know, the cognitive demand is changing all the time, and presumably the network activity and connectivity also has to change all the time to meet those demands. And so the idea is that if you don't have as much variability, you probably don't have as much flexibility in that ability to switch between networks. You know, what that means in terms of the underlying neurons, I think is pretty much anyone's guess, although there is some data, not from my lab, but from a group in Berlin, suggesting that if you give people a dopamine agonist, you increase bold variability, specifically in older adults. So it is somewhat related probably to changes in neurotransmitter systems. You know, but that's a very complicated question that we don't have good answers for at the moment, I'm afraid.
Sure. I have one more question, Cheryl, if I could. I was kind of trying to look at some of the scales on those plots you were showing to get a sense of really, if you like, the biological effect size here. I mean, clearly you showed some results that are statistically significantly different and others kind of weren't. But are we talking about, do you think, you know, effect sizes here that are of the order of like 1% or 2 or 5, or are they really very, very small effect sizes?
Oh, that's a good question. I mean, a lot of the graphs that I showed you were sort of things like brain scores from PLS and things like that, where it's, I guess, somewhat more difficult. In the univariate analysis, I mean, I guess the only thing we really haven't as an estimate of effect sizes would be the F, in the analysis of variance, and some of those were quite large. But to answer your question, I think I really don't know how large the effect size is. That's really an excellent question that we haven't really sort of delved into. I suspect, as with all things Fmri, it's not huge, because it's very rare to find anything in Fmri that's a huge effect size, but I don't know that for a fact.
Thanks, Cheryl.
Great, excellent. We have one question from the Q&A from Dragon. So have you ever used computational modeling of a behavior such as drift diffusion modeling to characterize the latent cognitive components? If so, how do they change with aging, and which network correlates with such changes? So that's a question from Dragon.
Yeah. If I understand the question correctly, it's asking about drift diffusion modeling. And the only time we've ever done anything really like that was a number of years ago. I had a postdoc who was interested in that kind of modeling. And honestly, I can't remember what the results were because it was quite a while ago now. But basically, we have not used that approach in anything that we have done recently.
Okay, good. So in view of time, we probably don't have time for further questions. Let's thank our speaker today. It's really excellent and very insightful. At least I learned a lot from today's presentation. Thanks a lot, Professor Grady, and thank you for your time.
Thank you.
So before everyone go, we have our next seminar coming up in June with Professor Alex Benito and Dr. Stuart Oldham from Toronto Institute and Monash. So looking forward to seeing everyone in June for next seminar. Thank you. Bye.